So if you look at the structures of the cell wall, it determines the cell shape. It prevents the lysis due to change in osmotic pressure, which we know about the plants. They are very good in terms of changing osmolarity. If you put a plant cell in a very dehydrated condition, and then after 3-4 hours if you put water, then the plant cell still get healthy and properly completely fine because of the cell wall they have. But if you do the same thing for an animal cell, they have only cell membrane. They can either burst the cell open or can shrink the cell down. So peptidoglycan is a primary component of a bacterial cell wall, while cellulose was the primary component for a plant cell wall. So that's the difference. Now the peptidoglycan has a unique macromolecular composed of repeating framework of long glycan chain cross-linked by short peptide fragments. Now the chemical nature of bacterial cell wall, you can see peptidoglycan is made up of two amino sugars, N-acetyl glucosamine, N-acetyl muramic acid. So we call it N-acetyl glucosamine or NAG, N-acetyl muramic acid or NAM. So this NAG and NAM are the two major amino sugar components of peptidoglycan layer, and you can see this is the NAG structure, this is the NAM structure, and both of them has a chain, and especially the NAM also have 4 amino acids linked with it, namely L-alanine, D-glutamic acid, D-diamino-pimelic acid, and D-alanine. 4 of these amino acid structures are linked to one another in that sequence, in that order, with the N-acetylmuramic acid, and both of them are connected by beta 1,4 linkage, and that forms this cell wall structure, this peptidoglycan complex structure of a gram positive bacteria. Also it's present in gram negative, but in gram negative the layer is very very thin, but in gram positive the peptidoglycan layer is very thick. So for a gram positive cell wall, it's near about 20 to 80 nanometer thick layer of peptidoglycan that is found. It includes teichoic acid and lipoteichoic acid. Function in the cell wall maintenance and enlargement during the cell division, and they move cations across the cell envelope, stimulate a specific immune response as well. So if you look at the zoomed in version of a gram positive cell wall, you can see this is the cell membrane inside and outside the cell wall, and for gram positive bacteria the cell wall is the outermost layer. In the outermost layer we see this NAG and NAM connected, they are cross-linked. Apart from that, the teichoic acid with red color chain, lipoteichoic acid with yellow color chain, and surface proteins with green colored are visible very clearly. So in this other picture also you can see the NAG with red and NAM with green, and lipoteichoic acid and teichoic acid are also provided. Now both teichoic and lipoteichoic acid's presence is very important in terms of maintaining the cell wall structure, in terms of maintaining the cell wall as well as expanding the cell wall while the bacterial cell is increasing in size before the cellular split or cell division. Some cells have a periplasmic space. The periplasmic space is the place between this peptidoglycan layer and the membrane. So the cell membrane, this one, and the peptidoglycan layer, there is a space between it that can be periplasmic space. The space is always present, but for some bacteria that space is little big, so we call that bacteria with a large periplasmic space. Otherwise that may be little small as well. For a gram negative cell wall, now if you think the gram negative cell wall, it has inner and outer membranes and a periplasmic space. Between them contains a very thin peptidoglycan layer. So think about it, the peptidoglycan layer is very thin, but there is a difference between inner and outer membrane, and there is also a periplasmic space must be present between the two. So the outermost membrane of gram negative bacteria is the most important structure of a gram negative bacteria. It's a signature structure of a gram negative bacteria. We call it a lipopolysaccharide layer or LPS layer. So lipid portion is known as endotoxin and may become toxic when released during infections, while may function as reporters and blocking immune responses. and they also contain porine proteins in upper layer that regulates the molecules entering and leaving the cell. So if you look at the picture very clearly now, the contrasting picture between the gram-positive cell wall and gram-negative cell wall. Gram-positive bacterial capsule, layer outside, and gram-negative bacterial capsule layer outside. So what we can see that in gram-positive we have this very thick peptidoglycan layer like a brick, and there are some this lipoteichoic acid and teichoic acid on the surface, and the membrane is in the bottom portion of the cell wall. There may or may not be periplasmic space. But in case of gram-negative, there must be periplasmic space whenever there is a gap between inner and outer membrane. So this is the innermost, this is the inner membrane, inner cell membrane. This is the outer cell membrane, and this is very thin brick-like peptidoglycan layer, and the gap between peptidoglycan and inner membrane is a periplasmic space, and the gap between peptidoglycan and outer cell membrane is another another periplasmic space. And in the outermost membrane, these are porine channels that are present, which allows entry and passage of different components in and across and out of the cell membrane, which is very, very important. In case of gram-positive, that's very difficult because there is a cell wall and there is no porine. But in case of gram-negative, this is mostly behaving like just an animal cell kind of situation, where there is only cell membrane and pores are there, channels are there through which any component can go in and out. So this is a contrast table between a gram-positive and a gram-negative bacteria. So number of major layers in gram-positive is only 1. In case of gram-negative, it's 2. Because actually it's not good for 1 and 2. I can say in gram-positive it's 2 because it's lipid cell membrane, inner cell membrane, and peptidoglycan. In case of gram-negative, it's inner cell membrane, peptidoglycan, and outer cell membrane. Chemical composition for gram-positive: peptidoglycan, teichoic and lipoteichoic acid, mycolic acid, and other polysaccharides. While in gram-negative, lipopolysaccharide, LPS, lipoprotein, peptidoglycan, and porine proteins on the surface. Overall thickness for gram-positive, it is thicker; for gram-negative, thinner. So gram-positive, the layer is 20 to 80 nanometer thickness. The peptidoglycan for gram-negative is only 8 to 11 nanometer thickness. The outer membrane is absent in gram-positive, while it is definitely present and has a signature characteristics of gram-negative bacteria. Periplasmic space is very narrow in gram-positive, while it's very extensive in gram-negative. The permeability to molecules for gram-positive is more penetrable, gram-negative is less penetrable. Although it's shocking to think, but yes, it is correct. Now the question is why and how we are calling them gram-positive, gram-negative. There's nothing to deal with the decimal system of gram, kilogram here. It's all about the name of the researcher who developed the staining method that help us to distinguish between the gram-positive and gram-negative bacterial cell. So it's a differential stain that distinguishes cell with gram-positive and gram-negative cell wall. So if it's a gram-positive, they retain crystal violet and stain purple in color, while for gram-negative, they lose the crystal violet because they don't have the thick peptidoglycan layer, and they are getting stained by the red saffronine color. So in case of gram staining, we use two different stains, crystal violet, which is known as CV, and saffronine. So CV and saffronine both are used, and there are also decolorizing agents are also used, moderants are also used. But if a cell contains and retains that purple color of crystal violet, that means that cell is gram-positive. And if a cell retains the counter stain or the stain which is used later of that staining process, saffronine, red color of saffronine, then that cell is gram-negative. So it's important basis of bacterial classification and identification, because the very first thing that we do when we are working with an unknown bacteria is we want to know whether that bacteria is gram-positive or gram-negative, because we know that if it's a gram-positive bacteria, there should be some properties associated with it, and if it's a gram-negative bacteria, then there should be some properties associated with it. Then we can continue with other dichotomous key and other modes of bacterial identification. Okay, we can also use a practical aid in diagnosing infection and also guiding drug treatment. So how exactly the Gram staining is done? Before understanding the steps of the Gram staining, let's first know the components of the Gram staining. Four components are used. Namely, crystal violet is the first one. It's acting as a primary stain, stains all the cell walls as a purple one. Second one is iodine, acts as a mordant, combines with crystal violet to create a large insoluble dark purple complex. Now the third one is alcohol, acting as a decolorizer, solubilize a gram-negative outer membrane and flushing iodine crystal violet complex out of the thin wall, and thick positive cell resist this decolorization. Now this is the most important step and most important component of this staining, the decolorizer, because the moment we put alcohol, the outermost layer of a gram-negative bacteria is washed out, is flushed out of that cell. And as the outermost layer is flushed out and the cell wall was very thin, that iodine crystal complex, the crystal violet complex, is also washed out. So for gram-negative bacteria, every component of crystal violet and that violet color is completely washed out after treating it with alcohol as a decolorizing agent. But for a gram-positive bacteria, even if we use decolorizer, even if we use alcohol, but it's not going to dissolve that outermost layer, because the gram-positive, they don't have any outermost cell membrane, they only have a thick peptidoglycan layer, and the alcohol cannot wash it out. Thus at the end we use safranin, and as we know that safranin stains all the cell walls pink, but as the cell wall of gram-positive is already violet, deep purple, dark purple, it's not going to take the color of safranin. Okay, but the cell membrane and cell wall of gram-negative is colorless, so they will take up this color and will be turned as pink or reddish. So these are the steps of gram staining. Now the first step is flooding with crystal violet for 1 minute, and then we rinse it, because we hope that both of them will take the color, and that's true, both of them takes the color, both of the cell wall take the color, and cell membrane also takes the color in case of the gram-negative, as you can see here. The second step, flood it with iodine for 1 minute and then rinse. Iodine will help in stabilizing the crystal violet complex, because iodine crystal violet forms a large complex that cannot be left and removed even after rinsing. So that's stabilized. For both it's stabilized. Now the third and most important step, rinse it with alcohol until no more purple color comes off from the slide. So, and then we also rinse. So you can see when we put alcohol, it will rinse off the outermost layer of the gram-negative bacteria, and thus also removing the crystal violet and iodine complex. So all the color components are left, so no color left for gram-negative, but color still left for gram-positive. And the last step, flood it with safranin and then again rinse and dry. So when you put safranin for gram-negative, which is by the way colorless by now, will take the color of safranin and now will be pink and red in color, while the one gram-positive will not take the color of safranin because it's already had the purple, already had the violet color, sorry. So as it already had the violet color, it will stay with the violet color, and thus we can say the gram-positive will contain the violet color from crystal violet, and the one gram-negative will get a red color from safranin. So till this point we've been talking about the cell wall component of bacteria. Now there are also the non-typical cell walls present. Some bacterial groups lack typical cell wall structure. Example, Mycobacterium and Nocardia. Gram-positive cell wall structure with lipid mycolic acid, which is also known as the Cod factor, are present. Pathogenicity and high degree of resistance to certain chemicals or dyes are there in cases where there is a modification of the cell wall. And also basis of acid-fast stain used to diagnose the infection caused by these microorganisms, because they don't have a typical peptidoglycan components in the cell wall. Instead, they have a mycolic acid, or which is also known as the Cod factor, on the surface, and they act as a completely different virulent factor. So they cannot be stained with, and they cannot be identified with the help of gram-positive, gram-negative, gram staining. What we need to do is we need to use a different staining known as acid-fast staining to find out whether the cell wall components are made up with mycolic acid or Cod factor or not. And there are some bacteria, they do not have any cell wall at all, like Mycoplasma. The cell wall is stabilized by sterols, like whatever. But mycoplasma is those infective agents without any cell wall, but equally virulent, equally dangerous in Okay, so we're done with the second lecture of microbiology about gram-positive and gram-negative bacteria, as well as about the gram staining. If you like this video, please hit the like button, share this video with your friends, and subscribe this channel so that you can get more and more like this microbiology lecture videos.